Both the circulatory system and the lymphatic system work together to allow the immune system to function properly. What it does, the circulatory system exerts pressure that causes the fluid to leak from the vessels. This fluid is called lymph. This moves about in the lymphatic vessels, as you can see the red line and the yellow line. So what it does, it collects and returns the interstitial fluid to the blood. So on its way, this fluid also collect uh, pathogens or antigen. As like you can see here, there is infection here. So this lymphatic system take up this bacteria along with it to the nearest lymphatic node for activation of immune response. So apart from that, this lymphatic system has also role in transportation of lipid from intestine to blood. Let's discuss the role of lymphatic system in immune response or immune cells in detail. Lymphocytes originate in the bone marrow. So these lymphoid tissues and organs are divided into two types, either primary or secondary, depending upon their functioning. So the primary lymphoid organs or tissues are those where the lymphocytes are developed and mature, like bone marrow, where B cells mature and develop, and thymus, where the T cells mature and develop. The secondary organs or tissues are those which are places for interaction of lymphocytes with the pathogens, uh, like spleen, tonsil, adenoid, some mucosal lining in the intestine, as well as in the lymphoid tissues, and they are basically sites which trap the antigen and allows interaction with the antigen presenting cells and lymphocytes proliferation. Let's discuss them one by one. Thymus is one of the primary lymphoid organs. It is located just above the heart, and it is composed of two lobes, a right lobe and a left lobe, and it's a site for T cell maturation. During the process of maturation, about 90 to 95% of the T cell die while acquiring the T cell receptor or marker in the cortex part of thymus. So this high rate of death of T cell is due to the fact that the immune system eliminates all those T cells which display the surface marker which can react to self antigen, and it also tries to eliminate all T cells which have surface marker which cannot react to any of the antigen. So this process makes the whole immune response highly specific. Lymph nodes are the secondary lymphoid tissues present at specific site on the body, and these are basically bean shaped, and they are site for interaction of the lymphocytes with the pathogens. The naive lymphocytes, basically naive means all those lymphocytes which have never been exposed to an antigen. They circulate between blood, lymph, and these lymphoid nodes. So the pathogen or microbe from an infected tissue are picked up by the lymphatic vessels and arrive at the closest lymph node. This lymph node filters the microbe and antigen and are site of interaction of phagocytic cell and dendritic cell. And you can see there are two types of cells of the adaptive immune response, like B cell and T cells. B cells are present mostly in the cortex region and T cell in the paracortex region. So when the antigen interact with B cell, they result in proliferation of the plasma cell and antibodies production. And when T cells are activated by APC, they promote the B cell immune response. So together they result in elimination of the pathogen. Spleen is the largest secondary lymphoid organ. which is present at the left abdominal cavity between the stomach and the diaphragm. So histologically, it is composed of two components, like the red pulp, and these dotted portions are the white pulp. The red pulp contains all the components of the circulating blood. However, the white pulp behaves or is similar to that of a lymphatic node. Unlike the lymph nodes, which are specialized to trap localized antigen from regional tissue space, the spleen is adapted to filter blood and trapping blood-borne antigens, and thus can respond to systemic infection. So its primary function is to filter blood and blood-borne antigens and microbes, and it is site for activation of lymphocytes to the blood-borne pathogens. As you can see, the cross-section of the white pulp region, this yellow region is the B cell corona, which is mostly populated by B cells, and when antigen comes, it activates B cells, which results in production of antibodies. And this blue region indicate uh, the region which is densely populated with the T cells. This is called periatrial lymphatic sheath or pulse. So once activated, T cell results in activation of B cell response. Another function of spleen is to remove damaged and old erythrocytes. There are some secondary lymphoid tissues which are associated with our mucosal membranes. These are called MALT, which stands for mucosal associated lymphoid tissue. These tissues are generally present at the primary site of entry of the pathogens, as we have discussed, which are generally the respiratory tract and the gastrointestinal tract. Walled or bronchial associated lymphoid tissues are associated with the respiratory tract, and GALT, also called gut associated lymphoid tissues, are associated with gastrointestinal tract. There are certain regions within these tissues which are aggregated, like in form of tonsils, which is present at the pharynx region, as well as adenoids, that is present in the upper part of the buccal cavity, and they perform the function of capturing the antigens which enter to the respiratory tract. And such one of such tissue is also present in the appendix, and there are specific patches called Pear patches which are present in the gastrointestinal lining, which are in form of a dome shape, and they have specialized cell called the M cells, through which the pathogen can directly transferred through the mucosal lining. So in the spare parts, there are both B cells and T cells, which then interact with the antigen and initiate immune response. Now let's see how these spare parts M cells actually work in our gastrointestinal
The gut
tract.
is often the body's first point of
With
contact
this
with microbes.
video.
Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients. However, these structures and the underlying tissues also host the body's largest population of immune cells. Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialised phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell, or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation.
As a part of last concept checkpoint, try to answer these two questions and we'll discuss their answers in our interactive session.